Welcome to the topic on genetics and pain. This is the way we're going to structure the topic. Go through a couple of introductory definitions. Then we're going to briefly look at the difference between the pain mechanistic genes and the pain susceptibility genes. That's quite an important differentiation to understand. And that'll lead us on into looking at how genetics affects analgesics that we use and the various systems that it affects. So a locus. When I started reading up on genetics and pain, I had to revise a number of these things just so I had an idea about what I was actually reading, and that's the reason for giving it to you. So a locus is a location on a chromosome with a genetic sequence, with a DNA sequence. An allele is a variant of the sequence at a different location, or at a given location. So same location to two chromosomes, and it's got a difference in the genetic sequence. So that's an allele. Now genome versus genotype. If you have a look at it, they're pretty much similar really. A complete set of DNA, which is a genome, and a genotype is the same thing, a full set of hereditary information. But the difference really is the genome is species specific, whereas a genotype is individual specific. So I have a particular genotype, and as a human, there is a particular human genome. And phenotype is the one thing that we all remember. That is the expression of our genotype, and phenotype is completely different, and that's dependent on a whole host of factors which we'll elaborate on shortly. This is the crux of the definitions that we need to really get to, and that's a single nucleotide polymorphism, or shortened to SNP, and it's a single nucleotide base variation that occurs, and then that can lead to a phenotypic change. So what you have here is the common one. It's that cytosine thymine nucleotide base switches or SNPs variations, and this in itself has implications. And this is the crux of all the pharmacogenetics. Is we're looking at SNPs, single nucleotide polymorphisms. So now let's compare pharmacogenetics and pharmacogenomics. They're essentially used interchangeably, and you could use them interchangeably. However, there are slight differences. So I've had a look deeper, and I've come up with the two main differences. Essentially what they both mean is that it's the science of examining inherited variations in genes that dictate how a drug works. So that's both of them, genetics and genomics. We're both looking at the same thing. However, if you look at pharmacogenetics, this is essentially looking at the SNP, so single nucleotide polymorphisms, and how those changes can affect modification of drugs and glucuronidation of drugs, so how drugs are metabolized. So that's pharmacogenetics. Whereas pharmacogenomics is a much grander definition, and you can think of it as looking at genomes on a much greater scale, so on a population-based scale, so looking at big groups of patients, groups of populations, looking for SNPs that are associated with particular diseases, and then working your way backwards. And the classic study that they do are called GWAS studies, so genome-wide association studies, and there's an example of a GWAS study. So genomics, wider, broader scale, genetics really focusing down on the SNPs and how they affect drugs and drug metabolism. So the aims of pharmacogenetics are essentially personalized medicine. That is what we're all aiming towards, giving the best drug at the best time, at the best dose, to give us the greatest efficacy with the least amount of side effects. And that essentially what is termed now personalized medicine. We're still a long way off, but it's something that's being focused on. So now let's look at pain sensitivity, and there are a whole host of features that can affect how we feel pain or how we're sensitive to pain. So this is having a look at the broader aspect really. There's genetic factors, environmental factors, and how they're related, and some aspects are completely genetically determined, some aspects are completely environmentally determined, and then anywhere in the middle there's a combination of genetic and environmental factors that can affect pain sensitivity. And the genetic factors are the actual genome and SNPs, gender, which is a whole different topic, and of course the ethnicity. And environmental factors are various things such as alcohol, drugs, medication, pollutants, and various diseases as well. And then of course pain sensitivity, specifically mood, cognitive set, and the context of the situation can have an effect on sensitivity of pain. So now let's start looking at the difference between mechanistic genes and susceptibility genes. Now, pain mechanistic genes are exactly what it says. They are genes that are vital to the mechanism of pain, to feel pain. Now, this is vital for survival. So if you don't have if you've got a problem with your pain mechanistic gene, you've got a problem with actually feeling pain, you're going to have a problem living and surviving. So pain mechanistic genes are relatively well protected. So small changes in pain mechanistic genes, small SNPs, don't necessarily translate to phenotypic changes or changes that affect the sensation of pain. That's good. However, it doesn't happen always, and there are a group of diseases where patients have problems with pain mechanistic diseases, and you'd expect them to not live far into adulthood, and that's exactly what happens in some groups of these patients. So the hereditary sensory and autonomic neuropathies, the HSANs, you've got 1 through 5, and we'll focus on HSAN 4, congenital insensitivity to pain and anhidrosis. And as you can see there's a problem with the NTRK1 gene, and that leads to a problem with an abnormality with nerve growth factor, the TRK receptors, and essentially the mechanisms of feeling pain. Now this group of patients, there are only a handful of these patients through the world. I think in the USA there's 17 or 20 patients. And these patients get into the news. So these patients become the focus of Oprah and other news features. And as you can see, CNN, BBC, these patients are out there, and they're the focus of a lot of attention. They don't feel pain. So it's something that the media loves. Now, pain susceptibility genes are something different. These are genes that have small changes in them, and that can change the way we feel pain. So we still feel pain, but we feel pain at different degrees and different variations. So these are genes that contribute to variations in responses to experimental painful stimuli and pathological conditions. And variations in these genes may lead to changes in pain sensitivity. And this is where all the genetic research is focused on, pain susceptibility genes. So looking at the SNPs and how they affect sensitivity of pain. Now there are a whole host of papers out there, and more coming out every week, looking at various SNPs and how they may affect pain susceptibility in the clinical situation or in the experimental situation. This is quite a good topic of, say, multiple choice questions. So looking at the compt gene, the compt enzyme, so catechol methyltransferase enzyme. And in the gene itself, a Val158 MET polymorphism has an influence on different responses to pain and stress and pain. So compt gene abnormalities can lead to changes in pain sensitivity. TRPV and OPRD1, so transient receptor potential vanilloid, and OPR delta 1, so opioid receptor delta 1. Variations in these receptors can lead to changes in nociception and heat sensitivity, and we've previously discussed that TRPV1 is amongst other things, a heat-activated receptor on primary afferent neurons. The CACNA1A gene affects calcium channels, and this is associated with familial hemiplegic migraine type 1. The ATP1A2 transporting glycoprotein is associated with familial hemiplegic migraine type 2. And the MC1R gene, so the melanocortin receptor gene, is associated with abnormalities in the K-opioid receptor, so kappa opioid receptor mediated analgesia, and this is the focus of quite a lot of research at present. And of course the MCR1 gene also carries the color for hair, so red hair, so there's an association with changes in susceptibility and possibly hair color as well, pain susceptibility that is, so a lot of focus of attention at the moment. Now let's move on to analgesics and how the analgesics are affected by SNPs. You can affect the metabolism of drugs, the transport of drugs across various membranes, how drugs interact with receptors, the neurotransmitters themselves, and of course sodium channels. So metabolism, let's go on to phase 1 metabolism. So phase 1 liver metabolism, the modification of drugs by redox reactions, and the commonest SNP that we know about is the CYP2D polymorphism. So CYP2D6 metabolizes certain drugs. If it's not there, if you have poor metabolizers, of which you have about 10% of Caucasians and only 1% of Chinese, if you have poor metabolizers, they don't metabolize the drug. So if the drug requires the drug itself to have an effect, that's good, you could have an effect. Whereas if the metabolism is required to activate the drug, and you have a poor metabolizer, you're not going to activate the drug, you're not going to have an effect of the drug. And the classic one, of course, is codeine, and codeine is dependent on CYP2D6. If you have a poor metabolizer, you're not going to metabolize codeine to the active component morphine, so codeine's not going to work. And that's why it doesn't feature as part of my analgesic regime. Whereas the ultra-rapid metabolizers are just the opposite. They're going to rapidly metabolize a drug. So if you need the parent compound to have an effect, it's not going to work because it's going to get metabolized really quickly. Or if you have the metabolite required, you're going to get quick conversion and possibility of effects and side effects. So codeine in ultra-rapid metabolizers, you're going to get side effects. Whereas the opposite could be said for another drug where you require the parent compound to be active. In poor metabolizers, you need lower doses. And an example of that would be the tricyclics. So tricyclics in poor metabolizers, you need small doses because it's the parent compound, that's the active compound. Whereas tricyclics, excuse me, whereas tricyclics in ultra-rapid metabolizers, you need a much higher dose. So for example, in a poor metabolizer, 5 milligrams of a tricyclic drug may have an effect, whereas you may need to give 150, 200, even 300 milligrams in an ultra-rapid metabolizer to have an effect because it gets so rapidly converted away. And of course, this is just one, CYP2D6. It has effects on multiple drugs, codeine, tramadol, tricyclics, venlafaxine, oxycodone, and of course methadone as well. And that's just one of the enzymes, and what we've got is a whole host of enzymes as well. So the crux of this slide is that it's complicated. There's a lot we don't know. Moving on to phase 2 metabolism, so the glucuronidation and the UGT system, of which there's UGT 1A and 2, of which there are 8 and 7 isoforms. So there in itself, you've got a whole host of SNPs in the second part of the metabolism that could interfere with drugs and the effects of drugs. And UGT2B7 is associated with the metabolism of morphine, hydromorphone, even some anti-inflammatories and anticonvulsants, and this, we don't know a lot about this yet. So again, growing, expanding, and this is probably why so many of the medications have such a variation in responses between our patients. So we've briefly discussed the metabolism of drugs, and now let's move on to the transport of drugs across biomembranes. So drugs are required to get transported across membranes before they can have an effect, or have an effect, say, in the central nervous system. So biomembranes, the blood-brain barrier is the classic biomembrane. the kidneys, the livers, even the intestine as well. So if you have abnormalities in biomembrane transporters, that could change drug availability or bioavailability. And the classic one is the ATP binding cassette superfamily, as it's so called, and the ABC transporter P glycoproteins, ABCB1. There are 50 or more SNPs that have been found. This is important in the blood-brain barrier. And drugs such as antidepressants and steroids are dependent on ABCB1 or this glycoprotein transporter before it can have its effect. So let's move on to receptors. Mu1 receptors, so the OPRM1 gene codes for mu1. Abnormalities with this gene could lead to abnormalities with mu1 receptor and hence problems with or differences in our responses to opioids. And the classic one, of which there's a lot of evidence out there, a lot of papers out there, you've got the references, is the A118G SNP, of which a couple of the papers, one paper said there were 15% of their study group had the A118G single nucleotide polymorphism. Another paper with 30% of the study group. And this SNP has been associated with abnormalities in pain scores, abnormalities in morphine and other opioid usage. So an example was, one of the papers was L-fentanyl usage, and interestingly associated with post-op nausea and vomiting as well. Now, a lot of papers have shown that these SNPs are associated with abnormal higher pain scores, excessive opioid use, and a reduction in the response to opioids as well. However, a recent meta-analysis had a look at these now numerous papers out there on A118G SNP, and they found, or their conclusion was, that there was no real evidence to suggest that there was an association between the A118 gene and significant abnormalities in pain stores. So the research that's coming out hasn't withheld the strict rigors of a first meta-analysis of its kind, but we'll wait to see, because I'm sure it will change with time. Now let's move on and have a look at neurotransmitters, and compt is the one neurotransmitter that comes up time and time again. It's a mechanism by which we catechol neurotransmitters are inactivated by adding in a methyl group. Now, depending on where you look, you'll find that low compt levels are associated with high pain experiences, and sometimes even vice versa. So the evidence out there is not consistent as well. But compt abnormalities, and I haven't said up or down, I've just said compt abnormalities have been associated with fibromyalgia syndrome as well. So fibromyalgia, a lot of studies out there looking at fibromyalgia, and we've discussed them in detail in the topic on fibromyalgia. But compt is in there, changes in diffuse noxious inhibitory control in there as well. Now let's move on to sodium channels. Good exam question, good MCQ question, a whole host of genes coding for various sodium channels. And it's the NAV 1.7, 1.8, and 1.9 are associated with pain and pain sensation. And it's the SCN9A gene that codes for the NAV 1.7 gene. Abnormalities in that gene, coding for abnormalities in that channel, could lead to problems with that channel, and hence the sensitivity of pain. Now, I just want to pause there for a second and go back to the HSANs, so hereditary sensory and autonomic neuropathies, and mention CIP, which is congenital insensitivity to pain. So the HSANs are one group where you would find an example of a congenital insensitivity to pain. And that's a wide group, and it's the HSAN4 group that we've mentioned before. So that's one example of a congenital insensitivity to pain. That's quite a severe example. They don't live very far into adulthood. But you may get some minor changes in the channels. So now we're talking about channelopathies as well, where you've got abnormalities with the NAV channels, and that can lead to various forms of congenital abnormalities or congenital insensitivities to pain. And this group of patients might live later on into life because the channelopathies may be slightly milder in their expression. So congenital insensitivity to pain is a broad umbrella term that includes channelopathies such as sodium channels and the hereditary sensory and autonomic neuropathies as well. Now, of course, that's an insensitivity to pain, but just on the flip side of that, you may have an excessive amount of pain. So if you've got a channelopathy that affects sodium channels, you may actually feel more pain as well. And this is another group of patients, and these are the erythromelalgia group of patients, or the paroxysmal extreme pain disorder patients. And there you've got some pictures of the patient with erythromelalgia. That has been asked in the exam previously as a short answer question, and it's one of those primary pain conditions that's probably important to have a bearing on. Now, there is a famous person, or so-called famous person, with congenital insensitivity to pain, and it's the blonde giant, as we know him from the recent Millennium trilogy. So the girl with the dragon tattoo, the girl who played with fire, the villain, this guy, has a congenital insensitivity to pain. Now, because he survived into adulthood, we have to only assume it wasn't an extreme form, so that one of the H-SANs, but it was probably a channelopathy that he had. That's the conclusion of this topic.